Thank you for the introduction. It's a great pleasure to be here and very kind of the organizers to invite me. I'm going to talk today about experimental models, which means our patients are about to get much, much smaller, like 1/500th to 1/1000th of what we've been talking about most of today. So we're going to be talking about newborn mice. And it's been titled Present and Future because we're talking about BPD then and next. And over the course of the last few years, I've had a lot of thoughts about whether or not we are modeling the correct thing in our animal models. And so today, I'm actually not going to show you any data. I'm just going to show you a lot of questions that I have. And I'm hoping that this will stimulate some thoughts and discussions amongst the community now, later, and in the near future. Because I think the time is ripe for us to reevaluate what we're doing with our animal models and whether or not we may be able to get our animal models to better mimic the diseases that we are trying to study, understand, and to treat. So in addition to this wonderful symposium today, Stephen Abman did a wonderful job of putting together a set of reviews in the Journal of Pediatrics, which came out over the past 4 months, and 3 of them that came out over the last 3 months spent a lot of time describing how important animal models were to understand the pathobiology of BPD and how they were going to play a role in us developing other management strategies and treatments for this disease. So what I'm trying to say to you are that the animal models are very, very important. And in the contribution from Christina and myself, we came up with a list of methodologies that we thought would impact the future of work on pathobiology of BPD. And amongst what we've highlighted as important is to develop refined animal models that may better mimic and recapitulate the pathological hallmarks of BPD that we see today. We think this is particularly important because Christina and I also itemized a couple of research priorities, and 9 of the 12 research priorities that we highlighted all were going to rely on animal models to make progress in how we understand the pathobiology of this disease or syndrome of diseases and potentially come up with new management strategies. So effectively the take-home message up until now is animal models are very important, and of course for them to be useful they need to do the job that they're supposed to do. They need to model the disease that we are interested in. So we've seen the slide before earlier. This is the course of lung development in many organisms, including humans and in mice. It proceeds more or less the same way, just a lot faster in mice. And I'm showing here some mouse lungs. Maybe I use the cursor. Mouse lungs, which demonstrate what lung development hopes to achieve. That is to convert a structure that looks like this into a structure like this, which has many small alveoli, generates a very large surface gas exchange surface area, and thereby can fulfill fixed laws which define proper diffusion. And when this goes awry, when the lung development doesn't work like it is supposed to do, I've borrowed some slides here from one of Alan Joby's reviews. We see the pathological hallmarks of the disease that we are trying to mimic in animal models, and that is alveoli that are larger and fewer in number, and this not only occurs in the immediate postnatal period, but also is persistent, at least in some cases, into adolescence. And so this is what we want to mimic in our animal models, and we do this a variety of ways. I do it in a hyperoxia chamber, expose newborn mice to elevated oxygen concentrations. Dick's talked a lot today about the mechanical ventilation models, and we've heard earlier today also about some larger animal models. And effectively what happens here is we are able to recapitulate the disease in that normal-looking lung would look like this, and we end up with a structure like this, which has larger alveoli and has fewer alveoli. So up until now, everything's great, and then a reviewer gets hold of your paper, and everything becomes not so great anymore. So this is kind of a selection of probably comments that come from a lot of people in this audience who review these papers. So for example, you use 85% O2, patients never see this. What are you doing? You don't know what you're doing. You exposed to hyperoxia for far too long, and review number 2 of the same paper, you exposed to hyperoxia for far too short. You've used 40% oxygen, but we've never seen any impact on lung structure, 40% oxygen. 2 papers later, why don't you use 40% oxygen? It would be much more clinically relevant. I have my suspicions. So we wean. Can't you do a gradient instead of just stopping it? Your insult's too stable. It's dynamically variable in the clinic. Can't you cycle it? You need to repeat everything in a different model of a different species. Hyperoxic model has no inflammatory component. Rory, I think you would disagree a lot with this. It certainly has a very strong inflammatory component. This review is wrong. I fail to see how exposing a mouse to hyperoxia will ever tell us anything about BPD. Well, there's my career down the toilet. And it's not just about animal models, it's also about how we measure the tissue. So I'm a bit of a pariah in the community because I promote stereology a lot. Don't you lose the effects? And even better, do you hold shares or financial interest in stereology software company? I don't, just in case anybody was wondering. And as irritating as many of these comments, and this is just a small selection of many of them, are in terms of revising a paper for resubmission and hopefully ultimately publication, I think that the comments are also instructive in that they indicate that the community in general is somehow dissatisfied with the models that we are using and how translationally relevant they may be. And I think that these are very, very important questions that we should be thinking about. I think there's a lot of truth in this. So to address this, I commissioned, or to start addressing this, a couple of reviews recently at the American Journal of Physiology, and some members of the audience were kind enough to provide us with their reviews, where they critically evaluated mouse, rat, rabbit, lamb, and baboon models. And once this came out, Amble and I then tried to read through them and synthesize what was some of the general themes, the concerns that are raised with some of these animal models. And among some of the things that came out, we heard earlier about how big isn't always better, but sometimes big is better. So increasing the size or evolutionary closeness to humans tended to be associated with a better translationally recognized model. And that was a good thing. Of course, our models have to be translational for them to be useful. But of course, that's not all good news, because the bigger the animal gets, the more expensive it gets. You have to do a lot of husbandry and instrumentation and husbandry also creates technical difficulties. And there are some perceived ethical concerns with using bigger animals. And in Germany, which is where my laboratory is based, we are absolutely not allowed to use apes for any kind of research. So the increase in translational or translationability comes with some negatives. Additionally, genetic manipulation, it's inverse here. It actually gets harder and harder as the animals get bigger. And some reviewers are very enthusiastic about telling you can do all of your experiments in rats and rabbits because you just use the CRISPR-Cas9 system and suddenly you can knock out any gene that you want. I've been fighting with one gene with CRISPR-Cas9 in a rat for 3 years now and I'm nowhere. So genetic manipulation is certainly harder as your animals get bigger and more complex. Although some elements of the technical difficulty go away. For example, trust me, trying to intubate a one-day-old mouse is much more difficult than trying to intubate a preterm baby. And this is not the case, for example, in lambs and apes. So there are pros and cons to all of the models that we are working with. What has emerged weighing up the pros and cons are three questions that I think are very important and that I think we should all be thinking about, those of us using preclinical models. One is how well do our prevailing models recapitulate the lung pathology that we see in BPD today? A second is how well do our injurious stimuli align with the pathological stimuli that cause the disease that we work with? And a third is how well do we measure various things in our lung material, for example, in lung tissue from our BPD animal models? Concerning the first point, there is some concern about do we know what we should be modeling? Many of these animal models were developed alongside the first clinical interest in BPD. And so a lot of the animal models were developed and then BPD got some attention and started to evolve into a different type of clinical entity, but a lot of the animal models didn't evolve alongside with it. So I think that one of the most important questions today is, do we actually know how the lung pathology looks in BPD patients? Because this is really what we should be modeling. Another concern, for example, the review who said use 40% oxygen because it's more clinically relevant. Well, is this even important? Because if you must remember, usually we're putting oxygen in the clinical setting into patients which have preterm born infants, they have immature lungs, they're very susceptible to oxygen injury. And whereas you get your mice, which are certainly born also in a saccular stage, but these lungs are competent for breath, the pups are not preterm, and they may respond very differently to these low concentrations of oxygen. And in terms of what should we be measuring and how should we measure things in the lungs, how really should we quantify lung structure? There's been a lot of developments in microscopy and the quantification of structural things in the lung. And a lot of this is not put into practice. We tend to rely on what we are already using, and there are now alternatives to a morphometrically assessed mean linear intercept or radial alveolar count, or a septal thickness that is measured just with a ruler on the screen of a computer. And Bizarrely enough, we pay almost no attention to lung function, particularly in the rodent models. We use lung structure assessments as our endpoints on pretty much everything. So I'm going to take the rodent models as an example to exemplify some of these issues, and I'm taking the rodent models for two reasons. One is they're probably the most commonly used models at BPD, but secondly, they're often our first-line models. So we find out something in a rodent, and then it looks promising, and so we then can justify the expense and the ethics in bringing this into a bigger organism, and hopefully, or maybe one day this ends up in the clinic. But if we get it wrong at the level of the rodent, we may get it wrong all the way down from there. So they're widely used, rodents, because they're small and easy to handle and comparatively cheap. Genetic manipulation is feasible, particularly in mice, and we say they're a perfect model because they're born in the saccular stage, but there are also disadvantages. So the lungs at birth are well adapted for gas exchange already. The infant, well, mouse pups are not preterm. Lungs are structurally distinct from ours, although I don't see this as a big limitation, and they're not easy to do anything interventional with. You can't get vein or a tracheal access very easily in newborn mouse pups. So if I would look at what some of the currently unresolved rodent issues in terms of using rodents to model BPD, certainly in terms of the hyperoxia model, issues surrounding how much oxygen to use and what duration and when you should apply the oxygen insult is a matter of interest. And a few other things which I will come to, such as mouse or rat, or which strain and how should we combine insults. And then there are some things, for example, Dick's talked a lot about ventilation already today, so I won't cover this again. A role of sex and litter size I won't cover at all, but as we know in BPD patients there is a sex bias. This is not necessarily the case in many animal models, and we should consider if this is relevant for our interpretations or not. Concerning what concentrations of oxygen to use and when to apply it, I think many people think that this is already fairly well standardized amongst people working with preclinical models, and this is not the case. So what you see here is a survey of the literature over a 2-year period where there are 41 oxygen injury protocols that are being used. And they span the gamut from 10% hyperoxia to 100% oxygen with or without LPS injection, usually before the pups are born, over quite a broad duration time period. And why this is important is because what's been highlighted in this review here is that in several instances, multiple instances, the same intervention has led to exactly opposite results using different oxygen injury protocols. So using a knockout, for example, some knockouts of antioxidant systems in the lung, using a pharmacological manipulation of extracellular matrix metabolism, BAPN, lysyl oxidase inhibitors, depending on which oxygen injury protocol you either get a curative effect of your intervention, no effect, or a deleterious effect. And if we consider that we're going to use this very often as our first line for these type of investigations to work out ultimately new management strategies for patients, try and identify candidate pathways for targeting and intervention, or the identification of lead compounds for a therapeutic pipeline, if we get it wrong here, then we've lost it. We may have missed the effect altogether, or we may think we're onto something when we are not. And once we start doing something, we like to finish it, and this could go for years before we finally throw in the towel. So I think that this is something that certainly warrants a lot of consideration. To look at this in a little bit more detail, if we consider, this is response to reviewers, by the way, all of these crazy interventions and oxygen injury protocols that we do. But if we look at a 40% exposure of pups from the day of birth until the end of 2 weeks of postnatal life, we can see that we get a reduction in alveolar number, get a reduction in the alveolar density, not much change in lung volume, no effect on septal thickness. And then we increase this to 60%, and we see more or less the same thing. We increase it to 85%. We see a more dramatic effect on alveolar number and alveolar density. And now suddenly we see a new pathological picture. We see an increase in the septal thickness. And oftentimes, when I review or when I edit, I see investigators justifying the use of the 85% oxygen because they want to recapitulate the two pathological hallmarks of BPD, alveolar simplification and increased septal thickness. Is this really true? Is increased septal thickness really something that we see in patients today? I don't think so. Nevertheless, this is why these exposure protocols are employed. And then moving on to some of the more eccentric suggestions of reviewers. Gradient reduction, we ended up doing a gradient from 85% to 21%, had no effect. The lung structure was fine, looked normal at the end of the 14-day period, which means when the high oxygen concentrations were hitting the lung, it wasn't really managing to block septation. And then we did this crazy oscillation, which was requested by a reviewer, which I still don't understand why, but we did it anyway. And that had some kind of intermediate effect, and I'm not quite sure why this is important, but I'm sure he's very satisfied that we did it. And then we looked at different windows of exposure, and of course, as you would expect, well, first of all, when you expose very early, from up to P3, which is really before the big burst of secondary septation happens, you don't really see an effect on alveoli number, density, septal thickness, or lung volume. But then when you start exposing over the period of peak, the bulk secondary septation in mice, or periods that span that, you start to see a more dramatic effect on the number of alveoli density, the septal wall thickness, although there are no changes on the lung volume. So we can certainly make a whole lot of different things happen in lung structure by doing different kinds of injury to the lung. But what do we really want to mimic? I think that's what we have to think about. What really mimics the pathology that we see today? These questions are not only important in terms of looking at the structure of the lung, but also looking at how interventions may affect lung development in this model. So I'm taking here an experiment where we have control mice maintained under normoxic conditions, and then we've used 60%, 85% oxygen. We've done an intervention with cottonseed oil, which is a mixture of 5 fatty acids. And as you can see here, under 60%, you can see that when we expose control mice to 60% versus 21%, we get a reduction in the number of alveoli. We treat with cottonseed oil, and we get those alveoli back again. But if we do this at 85%, we get a reduction in the number of alveoli, but we don't see any effect of the intervention. We've missed it. We've missed any possibility that this intervention is going to do something positive in lung development, because the insult is too strong. It's too strong for the intervention to be able to attenuate it. And clearly, something that we should keep in mind in the selection of our protocol is if we do something to very strongly insult the lung and phenocopy the histopathology that we see, we may not see the beneficial effect of a potential intervention. And in this sense, we then have missed something. Now, kind of the question that I really want to get here is, what should we be doing in animal models to mimic BPD in mice? So should we be mimicking the cause using the one review suggestion of 40% oxygen, because that's what we use in the clinic, but maybe this has no effect on the lung structure in our particular context? Or should we be mixing and matching a variety of causes that we would consider relevant insults in our patients today and use those to mimic the disease in our experimental animals? Or should we rather try and mimic the effects of these? So have a look at what is happening in the pathology of the lungs of affected patients today, and set this as our endpoints. This was not my selection. This was an error in my presentation, and now it seems to be some theatrical mask. Okay, it's not my doing though. I'm just noticing it now. So should we be trying to mimic alveolar simplification, septal structure? We've heard some very important work today, and I'm a big believer in how problems with the vasculature lead to problems in alveolar development. But very few models actually set their endpoint as an effect on the vessels. And we're totally ignoring lung function in almost all of the rodent models that we do, which I don't think is great, particularly if you consider the clinical definitions of BPD. Moving along, this is also not one of my symbols, but anyway. Now we're on the movies. That should be an is not equal to sign. And what we're trying to say here is that a mouse is not a rat. And this was a very interesting, one of my fellows, Philip Wright, did this editorial in response to some work that had been done looking at the potential utility of caffeine and other methylxanthines to drive lung development. So all of us know in the NICU we use caffeine and methylxanthines to manage apnea and prematurity, and there's always been a lot of interest about whether there's a knock-on benefit here on lung development, because it would be nice if the caffeine or the methylxanthines were also driving alveolarization. And what's very, very interesting here is that if you use a mouse, every study that's ever looked at caffeine or methylxanthines on lung development in a mouse showed that it either did nothing or it hurt the mouse's lungs. But a rat, a rabbit, and a monkey, suddenly everything gets good again. So if we're going to use a mouse here to look for lead compounds to manage the lung issues associated with BPD, perhaps a mouse is really a completely wrong model, and this would be better done in a rat, a rabbit, and a monkey. I know many of us think of a rat as just a big mouse, but it's certainly not the case in terms of the caffeine and methylxanthine function in lung development. So clearly, mouse or rat is an important consideration that we should also think about in terms of analyzing the effect of interventions on lung development in BPD models. And it's not just that a mouse is not necessarily a rat, but a mouse is also not necessarily a mouse. There are important mouse strain differences, which we often ignore, and the literature is terrible in reporting which mouse strains we use sometimes. But C57 Black 6 mice respond very differently to hyperoxy, for example, than do bulb C mice or FEBN mice. And it's not just the strains, but the substrains. So 6J versus a 6N has demonstrable differences in responses to hyperoxia, not just in the lung, by the way. There's papers out which show that the same is true of the development of the retina. So these are important considerations in the choice of an experimental animal that are often not thought about when these animals are employed in BPD models. Nevertheless, to move away a little bit from the models and more about how to measure lung structure, well, we all know that the lung gets bigger as the mouse gets bigger and the lung gets more complex, and eventually we want to end up with a lung structure that has a large number of small alveoli. And there's a lot of discussion, I think, now also about what are the best ways to measure these things, morphometry versus stereology. So the morphometric approaches usually employ paraffin-embedded tissues, but we select our sections there. Paraffin embedding can result in a lot of distortion of the lung structure. It's potentially biased because we select the tissues that we want. But these are very tried and tested techniques, and I think a huge amount of very valuable information has been gleaned using the mean linear intercept and the radial alveolar count approaches, and the measurement effectively by inspection of septal thickness. Stereology is something that is emerging. It's a pain in the ass, actually, but because it's slow and expensive, but it's very good at preserving the structure of the lungs, and it's because it's SUR, so systematic uniform random sampling, you can't be biased in what data that come out. And it can give you a lot of meaningful parameters: alveolar density, total alveoli number, your gas exchange surface area, an arithmetic mean, that means a septal wall thickness throughout the lung. And since we're talking about BPD then and next, I think stereology, we must at least think about using stereology as part of next, although this comes with, trust me, severe resistance on behalf of your lab members who don't want to put into practice this new technique that looks very scary at the beginning. And in response to the review comment I showed earlier, no, I don't hold a financial interest in these software companies that support stereology. Stereology gives you great data, very robust data. So for example, here you're looking at the increase in saccular or alveoli density over the course of lung development. You can see mice at 22 months developing a spontaneous emphysema here. You can see that the increase in density actually proceeds probably even beyond P28. Total number of alveoli, as you would expect, increase with increasing age, but a lot of this is commensurate with the expansion of the lungs, the growth of the lungs over time. And you can also see that the septal wall thickening is reducing over a very long period of time. It continues to thin up to P28 and perhaps beyond. And these are parameters that you can get out from stereological approaches that you will not get out from the classical morphometric approaches, but which also have a lot of important utility in what we do. You don't only have to use this for alveolar structures, you can use it for vessels. So for example, here I'm showing you that it's has extremely high resolving power. Looking at a particular knockout mouse we worked with, you can look at differences of less than 1 micron in the thickness of a vessel wall in developing mice, which this was also not the symbol I used, but looks very interesting. And it's important to know that this methodology that we use to study, to stereologically study lung structure and lung vessels can also be applied to airways, airway wall thicknesses. It's a very, very versatile technique, and I would strongly encourage consideration of its use by the community. There's also some interest in being able to quantify more things about capillaries. I think capillaries and how many and how long they are a key part of what happens in normal lung development and arrested lung development, and I'm very hopeful that there will be more work in the capillary analysis area from a stereological perspective in the coming years. Can the pain be lessened? So everybody hates me when I talk about stereology, so we are working in the lab about trying to lessen the pain. Can we do less? And perhaps we can. So we've done one study where we fragmented the lung, a mouse lung, into its 5 constituent lobes. And these lobes were nicely representative of the whole lung in terms of the effects of hyperoxia, but this is probably specific to the particular intervention and would probably have to be assessed separately for the different interventions that people may be trying to look at. And there are a lot of other things we can do. So Steve and Peter and others in Denver have done amazing work doing 3D reconstructions of human pathological material, and this is something that is now also being done in mouse models, which I think is very important. So you can assemble thousands of sections and end up walking through a precapillary arteriole, through the capillaries, and then into a postcapillary venule. And I think that this will also tell us a lot about what is happening in the vessels of affected lungs that has not been done before. So this is all part of the next in terms of BPD then and next in animal models. One very concerning thing about a lot of what we do, we often show changes in number of alveoli and being able to give a certain proportion of alveoli back, but almost nobody looks at whether this has any functional consequence on the gas exchange properties of these lungs. There is some work coming out. This is one that I've highlighted here. I think Amble's also done some in P14 mice. These had a BPD model, then let the mice grow to adulthood and looked at carbon monoxide diffusion. But I think that there needs to be more focus on functional studies in developing lungs and the interventions that we do in our BPD models. Examples being using whole body plethysmography and forced oscillation approaches. So these are my questions for you. As I told you, I was going to come up with lots of questions. So I think it would be very good if we would look at what we should be modeling in terms of BPD pathology and ask ourselves whether we really know what the pathology of these lungs look like today and how well this is recapitulated or mimicked in the animal models that we use. I don't think we really know this, and I think that we need to figure out ways to determine this. We have to consider what our endpoints should be. Should we focus more on structure or function? And if structure, what elements of the structure? And if function, what about the function? Should we try to mimic the clinical causes, such as particular concentrations and durations with and without different inflammatory stimuli? BPD is not just a lung disease. There are a lot of organs affected in these patients, and they're almost entirely ignored in animal models. There is some nice work coming out now, so I've seen a few people putting mice that were exposed to hyperoxia as neonates through mazes to look at cognitive ability. There's some studies on the brain and on the well, actually almost all of the extrapulmonary organs, but it's just one or two studies, and brain, eyes, heart, gut, diaphragm, and I'm a particular fan of placental issues, so I think that these are all things which would be nice to explore in animal models that haven't been explored before. And I'm a very big fan of the larger vertebrate models, and I think that there's been a move away from these, which I think is not good and not helpful. And if there's one thing I could change about the animal model world of BPD, it would that more people would be start doing these more technically complex and unfortunately more expensive models. But I think that those models where we can actually deliver something pre-term and we can ventilate something and get meaningful data out, I think it would be wonderful if we had more of these large animal models in place. And of course, how we look at our samples, particularly more three-dimensional reconsideration of the lung. A lot of preclinical studies only think of the lung in a two-dimensional way, which is really not great. So more three-dimensional consideration of the lung, I would say, is very much indicated. Before I stop there and invite any discussion if there is time, I'm actually going to take the moment here to do two bits of advertising. One of them, well, apart from thanking Claudio and Agnieszka for generating most of the data that I've showed you, this is part of the team at the San Francisco ATS 2016. One is the American Journal of Physiology, so I will take over from this man in January as editor-in-chief. Please send your papers to us. So we don't only do physiology, we actually do molecular biology. Some people sometimes very surprised by that. But we're also very interested in studies on clinical material. I mean, all neonate material, clinical material is rare, and if you do novel studies on that, we would definitely be interested to publish it, even though it is descriptive, so please consider the journal. In the audience, Kurt Albertine is also here. He has a wonderful journal, The Anatomical Record. It's also a great journal, so please consider these two journals for your work. It's not all about the blue journal, or the red journal. Okay. And additionally, I would like to raise something about the European Respiratory Society. I'm currently in the middle of my mandate as the research director of the ERS. It's a very big society of clinicians and basic scientists. But what a lot of you may not realize is we have a very active fellowship program that doesn't only look after Europeans. So at least 3 of our fellowships look at bringing a host to or taking a host from any institute in Europe or abroad, which includes the United States, for short-term fellowships, long-term research fellowships, and our Marie Curie Respire fellowships. So if you have a European fellow that you would like to bring to the United States, or if you have a fellow in the United States who would like to spend some time in Europe, please consider applying for these awards. They are ridiculously not well known and ridiculously easy to get. So if you fulfill all of the formal requirements, you stand between a 20% and a 50% chance of getting the award. And they are very generous awards. So if you have any questions about this, you can contact my office in Switzerland, or you can contact me, and I can put you in touch with the people in the Fellowships Directorate. But please consider using these facilities and submitting either to Kurt's journal or my journal. Yeah. Yeah. Thanks.